Our next challenge we will address is how do we insert our target gene of interest into another cell or organism. This can be achieved by a technique called gene cloning, which is either making identical copies or clones. That's why it's called gene cloning, okay? It makes clones of the original piece of DNA replicated within the bacterial cells, or introduce new DNA sequences in the cells to acquire and studies the gene products from that particular gene of interest. Now, before the development of PCR, this method of gene cloning was what scientists used to amplify a specific gene, which is, of course, a much longer and more tedious and labor-intensive method compared to PCR, because PCR, you're just throwing in the materials together, the DNA, the enzyme, DNTPs, let the machine run for a couple hours, and voila, you have your products. But for gene cloning, we're talking about days or even weeks of work, because you have to first purify that particular gene, put it into a cloning vector, and wait for cells to grow up, and then confirm presence of that particular gene of interest inside your cells. So it's a labor-intensive and tedious method, but we will go through the process still, because today we are still using this technique to make many copies of a specific plasmid and insert the plasmid into other cells, such as other bacteria or even eukaryotic cells, to study that particular gene's phenotype on the cells. So the first thing we need in gene cloning is what's called a cloning vector. Okay, a cloning vector is basically a circular piece of DNA molecule that we can design to have a foreign piece of DNA fragment attached to, so that we can introduce this particular fragment into another cell. A cloning vector then must have these three basic components. One being that they have to have an origin of replication somewhere within the plasmid, such as in this diagram here. So without origin of replication, you can't have the plasmid replicated, and your cells will never divide to have this particular plasmid. Okay, so in order for the DNA to be amplified within the host cell, you have to have the origin of replication. Second thing it has to have is selectable markers. So this, for example, here is a selectable marker. This will enable us to identify cells that has taken up the vector. Remember, one of the challenge questions we proposed in the beginning of the lecture was, how can we tell whether a cell has taken up our DNA of interest? Well, if there is a selectable marker, then there's a way to tell and select for cells that has this plasmid. Okay, usually the selectable marker is in form of an antibiotic or other genes that we'll discuss in a bit what selectable markers are doing. Okay. Third is that they have to have one or more unique restriction sites. So this is how we are able to insert our DNA of interest into the cloning vector itself. So that's why you see these cleavage restriction enzyme sites: HinD3, EcoR1, SAL1, BAMH1, PST1, etc. So those are different restriction enzyme sites on the cloning vector. Now, the thing about cloning vector is that you have to have only one restriction site for each restriction enzyme. You hardly find two HinD3 restriction sites in a cloning vector, or 3 or 4 EcoR1 sites. It's because when you have multiple restriction sites for the same restriction enzyme, then when you cut the plasmid, you end up cutting the cloning vector into tiny little pieces, and it's hard to put them back together in the correct order and arrangement. And hence, that's why cloning vectors are designed to have only one type of restriction enzyme, unique one. Okay, you can have multiple different restriction sites, but you can only have one unique one. Okay. All right, so these vectors can then be put into other bacterial cells by transformation, a method we have already discussed in the bacterial genome chapter and done in lab actually when we were working with P-Glow, the plasmid with GFP, the green fluorescent protein gene that makes the cell glow under UV light, and the plasmid with the LAC-Z gene. Okay, so some transformation can happen naturally on its own. Others, like the one we did in lab, require adding a chemical and change of temperature. Remember, we added calcium chloride to make the membrane around the bacteria more permeable to plasmid DNA in the lab. We also heat shock the bacteria by putting it on ice and then quickly in the warm water bath to heat shock the cells and allow easier uptake of the plasmid DNA itself, or cloning vector. In research lab, there's actually machines that does what's called electroporation, which uses electric shock to assist in plasmid uptake into the cell. Okay, and remember these nice little drawings you guys did with your P-Glow plasmid bacteria that you were able to plate on your agar plates. All right. So there are a couple different types of vectors that can be used for gene cloning. The most common one that's used all the time is called plasmid vector. This is a circular DNA molecule that exists naturally in bacteria, which we can actually harvest and alter to contain our gene of interest for amplification. Okay. Before we had PCR, this is how they used to amplify a particular gene, is by using these bacterial cells. So that's why they would use these plasmid vectors for that purpose, is just to amplify that particular piece of DNA using bacterial cells as our little machine that copies that particular gene of interest over and over again. Now, how can we insert our specific gene of interest into a plasmid vector is the next question. Like for P-Glow, bacteria don't have the GFP gene to make glowing proteins, so that the plasmid had a special gene insert, that has the GFP gene. Of course, we bought those plasmid in a lab kit from a company, but we can technically make it as well. It just takes a while and may fail if procedures were not followed correctly. And to save time, that's why we bought those kits that came with the plasmids already made with the specific sequence already inserted in there correctly for us. But technically, in a lab, you have to do it yourself. Okay, so the steps to insert a gene into a plasmid is: first, you will cut the foreign DNA containing gene of interest with a particular restriction enzyme. Now, you have to cut the plasmid with that same restriction enzyme so you're creating those cohesive ends. Remember the glue I was talking about earlier that sticks to each other because they have that single strand overhang. So in this case, in this picture as illustrated here, you have your plasmid and your foreign piece of DNA. We're going to cut both DNA with EcoR1 restriction enzyme. Okay, and EcoR1 will create these specific sticky ends, cohesive ends on both your foreign DNA and the plasmid DNA. Remember, this particular parts of the plasmid will now be complementary to this particular part of the foreign DNA, and this particular part will be complementary to that part of the foreign DNA. So now they will anneal together and bind with each other based on just normal complementary base pairing, and now your insert of DNA has become a part of the plasmid. Of course, you have to treat the plasmid with DNA ligase to seal these necks that's created by the cuts, so that you can permanently bind the sugar phosphate backbone together between the plasmid and your foreign DNA. Now your vector becomes a recombinant plasmid. So you have a continuous plasmid with your particular foreign DNA insert, and when you put this plasmid into a bacterial cell, because it has that origin of replication, the replication machinery within the bacteria will then replicate that particular piece of DNA plasmid for you because it has that origin of replication. It sounds very simple on paper, but in actual setting, you had to have a way of extracting and isolating and concentrating your gene of interest. Once it's inserted into the plasmid, you have to confirm it as well that it has successfully been inserted. Sometimes it's not just by selectable marker, but actually you have to do a PCR reaction to make sure your vector actually has, or your plasmid actually contain that particular sequence. This is especially critical in research when we're seeing that the cloning reaction didn't work in your cells. Then the next thing they go back to the clone, the vector, which is the plasmid vector, and PCR reaction with it to see whether it actually has your gene of interest or not. Now, a second type of vector commonly used is called an expression vector, which is similar to the plasmid vector, but it also contains additional sequences required for transcription and translation in bacterial cell. So for expression vectors, they are both for amplification and gene expression. Note that plasmid vectors do not have those additional sequences that enable the bacteria to do transcription and translation. So the only thing plasmid vectors are doing is allowing for the gene to be amplified within the cell. But with expression vector, you can actually ask the bacteria to transcribe the gene, translate the gene into an actual functional protein for you. In the case of pGlow, which is your GFP protein that the bacteria was asked to make with that pGlow plasmid. Okay? And still, these expression vectors has those three basic components of a plasmid vector. It still has your origin of replication right here. Okay, you still have your selectable marker. Okay, and then your particular gene of interest. Okay. Now, the other thing that we said it has to have is those different components for transcription and translation. So here's a segment that includes gene encoding a repressor that binds to your operator. We didn't talk about the operator region because that's part of prokaryotic transcription and translation, but you need to have this gene that sort of binds to the O operator region and regulate your promoter sequence within your bacterial plasmid in order to initiate the transcription and translation. And let's not forget, we need that special transcription initiation sequence as well, and also ribosome binding site. Remember that Shine-Delgarno sequence. Okay, so in order for prokaryotes or bacteria to recognize this plasmid as something that needs to be transcribed and translated, they have to have those particular sequences. And here are those restriction enzyme sites that, that is the base part of the basic components of a cloning vector. And of course, they still have those multiple restriction enzyme site within that area. And you're going to cut and be able to insert your gene of interest within that area of the plasmid. Okay? Same way. And sometimes these, special expression vectors need to be engineered for transferring a gene into eukaryotic cells, because for one thing, if we're talking about a protein that has to become functional with post-translational modification, then we can't do that in a normal bacteria, okay? Because bacteria don't do any post-transcriptional translational modification. So those kind of special proteins, then we would have to actually engineer different type of plasmid that would enable those things to happen to ensure the protein is functioning as it should in your eukaryotic cells. Okay. All right, so sometimes, we're not trying to transfect the gene and transform the gene in just bacterial cells, but sometimes we want to actually put genes into eukaryotic cells like yeast cells or even an organism that we'll talk about in a bit. So an example that we have used this type of expression vector to try to insert a particular gene of interest into eukaryotic cells was a gene that we have put with herbicide resistance for a crop plant that I was talking about in the beginning of the lecture, and also a gene that was to make clotting factor for a hemophiliac. Okay, so those are, eukaryotic cells we're talking about, plant cell and animal cells. So we're trying to put in this gene in eukaryotic cell in order for these things to be made. Okay. All right. So once the bacteria has been transformed with the appropriate cloning vector, we will then utilize its selectable marker to screen for cells that have successfully taken up the vector. So one selectable marker is usually an antibiotic-resistant gene, which is what you saw in the P-Glow lab. Remember, we had to use ampicillin-resistant, ampicillin-added agar plates to plate those bacteria. So if we include the antibiotic in the agar medium itself, only those bacteria with the antibiotic resistant gene within the plasmid will be able to grow. So that's, obstacle number one, to cut out those bacterial cells that didn't take up any plasmid at all. And we're only growing those bacteria that has taken up the plasmid. Now, sometimes a second selectable marker is used to ensure that the plasmid has taken up the plasmid that's taken up by the bacteria also for sure contain the targeted DNA sequence. How are we doing that? For example, a LAC-Z gene, okay, we also experience this in lab, but LAC-Z gene is often put into the plasmid as a second selectable marker. We also use this plasmid in our transformation lab just as a separate one from the P-Glow. Now, the LAC-Z gene encodes the enzyme beta-galactosidase, which actually cleaves a chemical called X-gal to produce a blue product. Okay, so this blue product will then make the bacterial colony blue in color. So what happens is usually the LAC-Z gene sequence will have a restriction enzyme site within it. Okay, so here's your first selectable marker with your ampicillin-resistant gene. This is the normal other parts of the plasmid. This is the LAC-Z gene. And it usually contains a restriction site where we can use the same restriction enzyme to cut the LAC-Z gene and our target foreign DNA sequence. So now we're particularly inserting our gene of interest into our LAC-Z gene, in a sense, interrupting our LAC-Z gene. Technically speaking, without the inserted foreign DNA sequence, you have your antibiotic resistant part of the plasmid, and then here's your other part that is your LAC-Z. Now, without the insertion, you have your whole gene sequence uninterrupted. The LAC-Z gene is intact, so you will make beta-galactosidase as it's supposed to be and produce bacteria that are blue. Okay, if your plasmid, okay, has successfully inserted this foreign piece of DNA interrupting your LAC-Z gene, then you will never be able to make beta-galactosidase in those particular types of bacteria, and hence those colonies will not look blue. Okay, so that's the readout goes for our second selectable marker. Okay, so bacterial cells with only intact plasmid, no recombinant DNA, which means our foreign DNA wasn't in there, will appear blue because the LAC-Z gene is intact. There is no interruption of a foreign piece of DNA. A lot of times, the integration of foreign targeted DNA sequence into the plasmid is not 100% efficient. We wish that was the case, but is not usually not. Okay, so in order to not do an extra step to purify for sure insertion plasmid, we're just putting it all on cloning them all into bacteria and use this second selectable marker to tell us which bacteria colony actually has the insertion sequence. Okay, So bacterial cells with plasmid that does contain your recombinant DNA or your foreign DNA will appear white because the LAC-Z gene was interrupted by your recombinant DNA sequence. Okay? So blue cells represent those that does not have your DNA of interest, your targeted DNA. It is your white colony that you want to pick from and grow a bunch of for your plasmid, because those represents the ones that actually has your targeted sequence. Okay? So this double selection scheme really cuts down the work required to evaluate the experiment. Of the hundreds of colonies of bacteria that ends up growing on your antibiotic resistant plate, only tens of them may turn out to be white compared to blue because, not, like I said, the efficiency of inserting that foreign DNA sequence is not 100% efficient. Usually then, after you have your tens of white colonies, usually then you may pick out a few of those white colonies, grow them in large batches in liquid media, for example, and then isolate and purify that plasmid DNA from these bacterial cells, and then analyze the plasmid further. Remember, I said you still have to confirm your plasmid does have that sequence even after these two double selectable marker, because you never know, just in case. Usually once we have a clone that has a good plasmid, we keep on going back to that clone to make more and more and grow more of that same bacteria with that correct clone. But if that correct clone was erroneous, or does not have something we want to begin with, then we're in trouble. We did months and months and months of studying this gene with this cloning technique, but yet, our original cloning vector was wrong. Okay, that's a whole month, waste of work and time. So it is very critical that we ensure the plasmid we extract from these bacterial cells actually include or have our DNA of interest or targeted DNA. Okay, and usually that can be done quickly through a restriction enzyme digest followed by gel electrophoresis to make sure that particular size of your foreign DNA sequence is there. Usually that's how we confirm that your plasmid is carrying the correct foreign DNA sequence. Now, if something goes wrong with your cloning, then sometimes people then go back to your cloning vector and actually do a PCR on it, okay, to make sure, or even sometimes sequence your plasmid to make sure that nothing went wrong with your cloning vector. So it is very, very critical that we're 100% sure that the gene we want is there before cloning the plasmid into other organisms or cells, or else things with whatever phenotype we are able to see may not be real. So it is very critical that we ensure that the insertion actually happened before we do anything with the plasmid itself. There's too many times in the lab where we've seen graduate students waste their time doing all these other experiments of analysis, the phenotype, but ends up, their original cloning vector had the wrong sequence in there, and they have to start all over again after months of work. So that's why this part of gene cloning is very critical. Again, I will upload an animation video on plasmid cloning to eCampus for you to visualize this process. All right. So before sequencing technology was developed, how did scientists figure out which DNA fragment have their specific gene of interest? They had to make what's called a DNA library. Okay, DNA library. It's a long process, but it's what they had to do back then. This approach is called the shotgun cloning method, because what they do is they will break your genomic DNA into many, many DNA fragments by using a whole variety of restriction enzymes. One of those fragments, or two or three of those fragments combined, should contain the entire gene of interest. So just like the shotgun, a large array of pellets will be sprayed in the direction of your target. At least one of those pellets within the hundreds of pellets that you spray will end up actually hitting the target. So that's why it's called a shotgun cloning method, because it's kind of like if I shoot a whole bunch of stuff out, one of them's got to hit the target, okay? So what they do with all the DNA fragments generated after cutting up the genomic DNA sequence is clone them into different plasmids, then transform the plasmid into the bacterial cells. They will search for the colony that actually carry their gene of interest using other screening methods. But, at first they would, create that genomic library or DNA library first. Okay, so here's a good diagram that illustrates this process. Let's say this entire thing is your genomic DNA, and we're going to cut at multiple different sites with different restriction enzymes we talked about. So now you have all these different little pieces of DNA that's been cut from those restriction sites. You have all of these DNA fragments. Let's say we're looking for this particular gene of interest. Well, back then there's no sequencing technique that tell us where our gene of interest is, so we can't design primers for amplifying our gene of interest. So we're just really cloning all of these little fragments into the bacteria cells, okay, via these cloning vectors. Hopefully one of them would actually contain our entire gene of interest, or sometimes maybe 2 or 3 of cloning vectors may have pieces of your gene of interest. Like, for example, this DNA fragment will have a tiny piece of that gene. This DNA fragment will have the other part of that gene, and both of these cloning vectors, combined together will give you the entire gene. So it's not a very efficient technique, but that's what they had to deal with before we can sequence the entire sequence of our genome letter by letter. Now, there are 2 different types of DNA libraries that can be made. One is called the genomic library, which is a collection of bacterial clones that contains all possible DNA sequences and fragments within the human genome. It is created from the original genomic DNA, broken into your fragments, and then cloned into bacteria here and grow on a plate, and we keep those clones separately in some kind of holding medium to put them in the freezer with. Okay, cDNA library is a different type of library where it is a collection of bacterial clones that only contain DNA sequences that are transcribed into mRNA, which means we're just looking at things that actually get transcribed, not the entire genome. I guess this is a way to narrow down the amount of DNA fragments we have to look for and sort of increase the chance that we're going to hit something that actually encodes a gene or actually be transcribed into a protein, which is usually the case because we're trying to study a particular gene of interest and the protein that it makes in the end. So cDNA library is sort of more directed way to save time and just look at mRNA sequences versus the entire genomic sequences. So the cDNA library is usually created by harvesting mRNA molecules, okay, and they will actually turn those mRNA molecules back into DNA via reverse transcriptase and then clone those DNA into the bacteria. That's why we call it cDNA library. Remember, we talked about when we turn RNA back into DNA using reverse transcriptase, we call that a cDNA. Okay, we talked about that earlier in another chapter. So here are the steps to create these libraries. Okay, so the first step is that we would extract DNA from cells, okay, and treat with restriction enzymes like I talked about. And these DNA fragments will be put into cloning vectors, and the vectors will be transformed into your bacteria. And now you have a plate of bacteria colonies, and each colony should contain different cloning vector that has your different parts of your gene of interest. Hopefully one of them would have your entire gene of interest. Now then, of course, you have to actually create a screening technique that can screen your clones, probably hundreds and hundreds of clones, to look for your particular bacteria colony that has your gene of interest in it. Usually they use an antibody or a similar gene from another organism and use it as probe to look for that particular bacteria with your gene of interest. Okay, so You can imagine how cumbersome this technique is, and you have to create this whole genomic library for every new organism you use to look for a gene of interest. But that's why it was called clone first, search later method, and why it's called shotgun cloning thing, because there's no other way to sequence your particular gene of interest. So hence, developing a method for DNA sequencing became very important in genetics to speed up the process of studying a gene. So that's why another scientist that discovered the first method to sequence a genome also ended up with a Nobel Prize. So the very first sequencing method was actually developed by another biochemist, Frederick Sanger, in the mid-1970s, which he won a Nobel Prize for in chemistry. He was actually one of the two known people that won the Nobel Prize twice in the same category. His other Nobel Prize he won was for the structure of insulin. So, and then of course his second Nobel Prize was for his particular sequence method. So this DNA sequencing method was called the dideoxy sequencing, or known as a Sanger method, after his name. So what's special about this method is that it uses a special nucleotide called DDNTP, or dideoxyribonucleoside triphosphate. What's special about this particular type of nucleotide is that they lack a 3' OH or hydroxyl group. So you see, this is your normal DNTP with that free 3' hydroxyl group, okay, which is what your DNA polymerase would recognize to add to your DNA template, okay? Well, with these DDNTPs, they don't have that 3' hydroxyl group, which means if this particular DDNTP was incorporated into the sequence, then your DNA polymerase would stop, because now it does not have a free 3' hydroxyl group to add the next nucleotide to. Because technically speaking, what happens is, here's your template A, T, T, G, something like that, okay? And then your DNA polymerase, let's say, adds a T, adds a A, okay? All of these addition of DNTPs has that 3' hydroxyl group, okay? 3' hydroxyl group. And that's what your DNA polymerase will recognize to add the next nucleotide in there. Well, let's say it adds a DD at the adenine, okay? This particular adenine does not have a 3' hydroxyl group. Well, DNA polymerase comes in and say, hey, there's no 3' hydroxyl group. So it stops, okay? It basically terminates a replication when an incorporation of DDNTP happens. So this is the basis of how this particular Sanger method of DNA sequencing works. So how is this used for DNA sequencing? Let me erase that. Okay. So here are the steps of how this is used for DNA sequencing. We first target your target DNA will be amplified using PCR, because again, trying to sequence a single piece of DNA is not possible at that moment. Okay, I'll talk about the steps now. Nowadays, we can actually sequence a single piece of DNA molecule, but back then this wasn't possible. So you still have to do PCR first of your target sequence that you want to sequence. And then you would add primers that are complementary to one end of your DNA. So here's your DNA template of sequence that you want to sequence, and you're adding a primer in there so that you have a 3' hydroxyl group to start the DNA synthesis process. And then you add all the 4 components of your DNTPs, okay, along with small amounts of DDATP, DDCTP, DDGTP, or DDATTP. So you're going to separate it into 4 different samples, 4 different test tubes, and in each of those, you're going to add DDATP, DDCTP, etc. And whenever the DNA synthesis incorporates one of those DDATPs, then it will stop, okay? And it will give you a fragment, okay? So DNA polymerase will be added to do the synthesis process. Now, the contents of the 4 tubes, once the synthesis is over, you will run the samples, these 4 tubes, on the gel electrophoresis. Now, what happened, what you're seeing here is that, let's say, of course, when the DNA polymerase is adding sequences complementary to your template, it's going to grab either a normal DATP or a modified DDATP, okay? So some cases, maybe the very first A, it's going to add in that particular DDATP and stops. So, and you end up with this very, very tiny little DNA fragment. But if it adds a DDATP later on in the sequence, okay, wherever there's an A, you can end up with different fragments of DNA sequences. Same thing with C's, G's, and T's. It's going to add in a random DDNTP somewhere along the way, stops a DNA synthesis, and create a shorter DNA fragment than your template DNA, okay? Now, it sounds confusing, but the readout is going to be very simple, okay? And usually either the primer or the DNTPs are labeled radioactively so that once you run your gels, you will be able to know which is your newly synthesized DNA based on the radioactivity labeling, okay? All right, so the concept of coming up with this method, again, like I said, it seems confusing, but the readout is very simple. What you will do first is when you run the gel, let's say you put all those 4 tubes into 4 wells that you will end up with lane 1, 2, 3, and 4. Lane 1 represents DDATP, lane 2 represents DDCTP, etc. Okay? And now you're going to look for the band that's closest to the bottom of the gel, which means it is the shortest DNA fragment you can have in the 4 sample. In this case, we have this very first band closest to the bottom that corresponds to G, DDGTP, okay? So that most likely mean the very first nucleotide was a G, because that's created the shortest DNA fragment possible, which is basically just the primer, okay, and your G, okay, this one right here. Now, you're going to look for the next band up in size, which is this one right here. This is the next band that's next in size. And in this case, this turns out to be corresponds with your DDATP. Okay, so that means the second nucleotide in sequence is an A, because this particular DNA fragment is what created this band right here on your gel, including the G nucleotide and the A nucleotide. And you're going to go the third band, the fourth band, the fifth band, sixth band, etc. Okay, so third band, fourth band, fifth band, sixth band. So those corresponds to the next nucleotide in sequence sequentially. Okay, that's why I said reading the results is not that confusing. It's very simple. You just look for the banding size and go up from the smallest band to the largest band, and the sequence is simply read from the very bottom of the gel to the top of the gel. From the bottom of the gel is the 5' end. So that means this is the 5' end. And then the top of the gel, the very last sequence, is the 3' end. Now, remember that this sequence is newly synthesized strand, and hence your original sequence should be the complement of this result. Okay, so here, this 5' to 3' sequence we're reading according to our DNA gel electrophoresis results, but this is what your DNA polymerase has added across your DNA template. So that's why the complement of the sequence would be then your original actual template sequence. So that's why this next to it is the actual sequence of your original template strand, which starts with 5' and then TCA, GCT, CGA, ATC. Okay, so I might include one of these problems in the homework assignment, so make sure you understand how to read this, which is fairly simple. Like I said, you're just reading the gel from the most bottom band to the highest top band on the gel. All right. And also, I will upload an animation video of this dideoxy sequencing technique to eCampus for you to visualize how this works, animated style. Now, today, doing it by gel and manually with hands will be too inefficient if we're talking about sequencing billions of nucleotides. I mean, just us counting 10 nucleotides took a while. Imagine if someone has to sit there and count 3 billion of those bands. That would take ages. So there are now automated machines, that carry out these dideoxy sequencing by automated programs. They will not require running a horizontal gels or reading of bands one by one anymore. Instead, it uses fluorescent dyes and laser scanners as detectors for the different DDA-NTPs that's incorporated into the sequence. This type of what they call first generation sequencing is kind of like iPhone. This is the first generation of sequencing. That means there's going to be second generation and third generation as well. So this is the first generation sequencing, which they still use the Sanger method with DDNTPs. So they're still using those DDNTPs, but now where they're going to attach a different color of fluorescent dye to these DDNTPs, then they will add these different dye DDNTPs to your DNA that need to be sequenced all in one single tube. So here's your single tube of your template DNA in there. And then they're going to add different DDNTPs, DDATP, DDTTP, and they're going to have a different colored fluorescent dye attached so that the machine can detect which nucleotide is it reading going across the laser beam, So the DNA will not be run through a horizontal gel, flat, taking up space. This is an automated, space-efficient kind of machine. So it uses something called capillary gel electrophoresis, where the gel is actually inside a small, tiny little tube that comes out of the machine. So as the DNA fragments are being put into these capillary gel electrophoresis, it will pass through these tubes and separate by size, and a laser will scan for passing of each DNA fragment by color to know which DDNTP it is that terminated the sequence. So this is why when you look up a Google image of sequencing, a lot of times you may see these readouts of colorful peaks like this right here. It is, what's generated from this automated dideoxy sequencing technique. And with this method, about 50,000 to 60,000 base pairs can be sequenced in a few hours. But that's still, kind of long ways from trying to sequence your entire genome, which is 3 billion base pairs. But it is already a big leap in efficiency compared to doing it by hand and manually by looking at the band size on the gel. So again, this is your laser beam. And then different DNA fragments will be run through this capillary tube-shaped electrophoresis gel, and then this laser will then read what colors are coming through the fragments, and then it's going to record and tell you what sequence it is in the DNA fragments coming one after another. Just automated way of reading the results. No, so like I said, just like iPhones from first generation to 10th generation, DNA sequencing also went from first generation to now third generation. Now, the first generation, which relied on Sanger sequencing, required probably hundreds of scientists. Like I said, you're sitting there reading those bands one by one, And when it became automated, you still require hundreds of machines to just read billions of base pairs, It costs a lot of money because it required so many people and so much machine, around $3 billion just to read a genome. And the time it took to sequence the entire genome would be 10 years, quite a long time. while second generation developed between 2006 and 2010. And this only required about 1 to 2 scientists. What a big jump, okay? And, it only required one machine. It's still quite expensive, anywhere from $5,000 to $10,000 per genome. And it takes about 2 weeks, but that's still a big jump from first generation. Okay, kind of like going from your first generation iPhone to now your generation 10 iPhone. Looks very different from back then. And then now to the third generation, we call the next generation sequencing between 2010 and 2015. Again, you only need 1 or 2 scientists, only one machine, but the cost has went down to be about $1,000. And within hours, you're going to get your whole entire human genome, all 3 billion base pairs of it, completely sequenced. I will actually upload a video on eCampus to show you how Helicos, one of those companies that does this, what is their idea and how it works. Is actually quite ingenious. Okay, all right, so we won't talk about all those different methods, but I will just wrap up sequencing with the second generation. The second generation of sequencing no longer uses a Sanger method. Only the first generation of sequencing technique relies on the Sanger method of using those dideoxynucleotides. The second generation on actually uses a method that detects flashes of light. Okay, and then second, third generation, all is about the same idea of relying on detecting flashes of light as the nucleotide is added to the DNA fragment. So now we're not looking for where it's terminating. Okay, we're not relying on a gel electrophoresis to separate the fragments after the DNA has already been made. We're going to look at the DNA synthesis as it happens. As that nucleotide is being made, added by your DNA polymerase, is going to generate that flash of light that correlates with that particular nucleotide, and it will be recorded and detected on the machine's detector, of course. Okay, and this particular second generation of DNA sequencing was called pyro sequencing because it relies on a chemical reaction that uses pyrophosphate to generate a flash of light that's detected by the machine. Now, how this works is that you have this method that allows for sequencing of many DNA fragments at the same time because it uses a special type of bead that serves as a platform to hold DNA. So again, that's another common theme with these new methods of sequencing. Not only is it relying on flashes of light that's generated as the nucleotide is added, but it also has some kind of platform that allows DNA fragments to attach to that tiny little platform that allows for you to look at multiple platform, which means hundreds and millions of DNA fragments all at the same time within that one little thing you put into the machine, okay? So in this pyro sequencing case, you're looking at beads, and each bead with different DNA attached can be placed in some kind of plate that has 96 holes in it, or we call it a 96-well plate. So with one plate, you can have 96 slots for 96 different beads with different DNA fragments attached. So the automated machine will allow 4 different solutions containing different nucleotide one at a time to be added, and they look for that flash of light generated when the nucleotide is added to the sequence. So, for example, here's your target DNA sequence you're trying to sequence. They're going to break it up into fragments, attach these adapters to it so you can prime the DNA synthesis reaction and also attach it to this spherical bead. So you have some platform to hold multiple pieces of DNA strands like this. And this bead full of DNA multiple strands then gets put into one of these wells within the 96 well plate. Okay, and then this plate will then be placed into the machine, and the machine will do its thing. Okay, what it will do, it will actually take turns flowing, putting solution that flows over the plate. First solution will contain just that one specific DNTP, for example, DATP. And then they're going to look for and record those flashes of light wherever adenine was added. Okay. Next, they're going to flow a second DNTP reaction, which is DCTP, DGTP, DTTP, etc., and look for those flashes of light. If there's no flashes of light detected, that means there's no C's. If there's no flashes of light when DGTP is added, then there's no G. Okay. And they keep repeating these flow of your 4 nucleotides, okay, until you're done with your entire sequence. If the next sequence was actually TT, okay, then you will have a higher peak of light that's generated, because if you have 2 T's that's added, you will have 2 flashes of light. So that's equivalent to a higher signal when you have continuous of the same nucleotide added. So far the sequence is ATT, okay? And then it will continue the readout as it continue reading the template sequence. And hence, this is how if you put in multiple plates can be used to read millions of fragments simultaneously with this method. Well, for the third generation, this is just a second generation. For the third generation, we now can sequence the entire person's billions of base pair within few hours for $1,000. Like I said, I will upload a video from Helicose Company. It's really amazing and ingenious how they figured it out to work, but that's where we are in terms of DNA sequencing. We went from taking 10 years to sequencing someone's genome to now being able to sequence someone's genome, all 3 billion base pairs of it, in just mere hours. And this is how now we can actually look for what we call SNPs, okay? Earlier, those single nucleotide polymorphisms that has association with heart disease or higher risk for heart problems or other brain problems. That's how we can look for those SNPs, is because of these sequencing techniques. Okay. All right. Okay. So when we discussed the idea of PCR, I mentioned the idea of using it for criminal investigations and settling paternity disputes. So how is this done? This is another of my favorite subject in genetics and science, because DNA fingerprinting is an area of forensic science. Okay, DNA fingerprinting, or so-called DNA profiling, is the use of specific sequences from a person's genome as a way to identify different individuals. It is based on the usage of what's called short tandem repeats, or STRs. Now, these STRs are repeated sequences found in many loci throughout the human genome, but each individual can have different numbers of repeats at the specific loci. So for example, here's individual 1 and here's individual 2. We have our genomic DNA that's been collected from these 2 individuals, usually through a swab of the inside of your cheeks. And hence that's why we were going to do cheek cells, DNA isolation in a lab setting. Now, this person can have 8 repeats of this particular STR, which contains just cytosine and adenine. Whereas this person can have 2 repeats for that particular sequence within this particular loci or position in the chromosome. So in this particular position in the chromosome, individual 1 has 8 repeats of this CA sequence. For individual 2 in this particular loci in that chromosome, they have only 2 repeats. So that's how we are able to generate different sizes of repeats that distinguishes different individual. The FBI actually uses the system of 13 STR loci that it helps, that mix up what's called the CODIS, which is Combined DNA Index System, to make sure we can identify past criminals with, sort of evidence that's collected from the crime scene. So here's the process of how a DNA profile is collected. So first, primers are tagged with fluorescent labels that are used to PCR the suspect's DNA. Remember I talked about how when you collect evidence from a crime scene, usually it's just a few DNA molecules, and we can never see that as a readout. So we have to PCR that particular piece of evidence DNA so we can amplify it, be able to read out on your gel. Okay, DNA fragments will be separated by capillary electrophoresis, and then your fragment sizes will be calculated. Now, the fragment are represented as peaks instead. Okay, here they're showing it in terms of a gel. Nowadays no one do these horizontal gel anymore because they're so time consuming and not so efficient. So everything is automated with a machine, and these uses these capillary electrophoresis, and those instead of a gel band readout, we don't even require dyes. They would actually show up as graphs for you, where there will be peaks showing on the graph, and homozygotes for STR allele will have a single tall peak, whereas heterozygotes will have two shorter peaks. Now, just using 13 short tandem repeat loci creates a unique pattern for each individual. No two persons on Earth ever will have the same pattern, just like fingerprint. Okay, so that's why it's called DNA fingerprinting, because no two persons DNA profile would be exactly the same unless they are identical twins, meaning they have the exact same DNA profile, DNA sequences. Okay, so here's a sample readout of a graph from a DNA fingerprinting of just 4 STR loci. Okay, so this is one loci, second loci, third, and fourth. This was a homozygous of that particular loci at DAS1179. Okay, so that was what you did in lab last week when you were looking at the CODIS frequencies, and you were calculating those frequencies. And this is the second loci of 2 heterozygotes allele having 2 peaks, and the fourth loci with that single homozygous peak. Now, how can we tell whether sequences match from the crime scene, evidence collected from the crime scene? So here's an example. You have suspect 1, which gives this particular pattern of these 4 loci, suspect 2, and then you have sample from the crime scene. So the sample from the crime scene gives you this particular pattern of peaks. So you're just going to look for that suspect with that exact same pattern to know which is the actual criminal. Okay, so in this case, is this suspect 1 or suspect 2 that has the same DNA pattern as the sample collected from the crime scene? Suspect 2, right? Suspect 2 has the exact same peaks at these 4 loci compared to the sample or evidence collected from the crime scene. And that's how we can match a person's DNA profile with whatever's left in the suspect crime scene. Okay? All right. Usually, so today actually, DNA fingerprinting is used in forensic investigations, assess paternity. For example, we can actually look for how likely is this child, actual child from this particular father. Let's say there's 2 fathers claiming to be this child's father. How can we find out which is the actual father? Well, this is what we're going to look at. We're going to look at the DNA profile of the child compared with the DNA profile of the mom and the dad. Okay, technically speaking, the child should share a lot of the peaks with the actual biological father. So that's why usually DNA analysis for paternity gives you a percentage. How likely is it that this child is an actual biological child of this particular man or woman? Okay. Study genetic relationships, identify specific strains of pathogenic bacteria, or even identify human remains. For example, if all you have left is a bunch of bones inside the soil, that you can never identify that person's from the dead body because you no longer have tissues. It's just bones within the soil. But we can actually take DNA sample from bones or the teeth of that bone and analyze it with this DNA fingerprinting technique and hopefully be able to find out who has that, has that matching DNA sequence with the bones that's found in the soil or the ground. Okay. All right. So usually the last process of studying a gene's genotypic effect fully is moving on to inserting the gene into an actual living animal like a mouse in order for the concept to be applicable to a human being in clinical trials. We call this moving from in vitro studies, meaning in a lab or in a petri dish plate, or to in vivo studies. In vivo meaning in an actual living organisms. Hence, scientists go from putting cloning vector to bacterial cells to putting our gene of interest into a living organism. We call these transgenic organisms. All right, so this is an example of reverse genetics, meaning that we go from genotype to phenotype. We can go from a gene of interest that we know the sequence for, and then we can find out what phenotypes does it give, okay, by putting that particular gene of interest into a different organism and observing what kind of effects we're going to have in that particular organism. Now, foreign DNA inserted into the organism is usually referred to as the transgene. Okay, and the mouse are the most often used model organism for human genes because mouse actually share 99% of genes with us humans. So that's why the next closest thing we can sort of test for human genes is the mouse because we have the most common shared genes. Now, how is a transgenic mouse created? How do you create one? Okay, so first you start with a female and a male mice, okay, and immediately after mating, the fertilized eggs are removed from the female mouse, okay. Then DNA will be injected directly into one of the pronuclei of the embryo, because usually when the sperm and the egg fertilizes, you have two pronuclei, one's coming from the egg, one's coming from the sperm. So you're going to inject those particular whatever gene of interest you want to inject using this, sterilized, very tiny glass needle into that particular pronuclei. And we can create either knockout mouse or knock-in mouse, okay? Knockout mice usually means mice that have a gene that's been knocked out of it, meaning that we have made a gene no longer functional, okay? So let's say we're studying, okay, what happens in a mouse without this particular gene? What are the phenotypic effects? Well, that's a knockout mouse. Knock-in mice is when we actually knock in a particular gene of interest we want to study in a mouse. For example, we want to study whether putting this gene in humans would actually be beneficial to a diabetic mouse. Okay, well, that's a knock-in mouse. Okay, we're knocking in something extra into the mouse's genome. Okay, so usually only a few embryos would actually integrate that foreign piece of DNA into the pronuclei genome, okay? And usually this is accomplished on its own via non-homologous recombination. And out of these embryos that are injected with the DNA, usually only 10 to 30% do survive at the end. So not a very high efficiency rate and not a very high survival rate. So these are not easy experiments we're talking about. Usually transgenic animals take anywhere from 1 year to 2 years to develop, okay? Embryos that then, once we know that that particular embryo survived and does have our gene of interest, they will be implanted back into the surrogate mother or the pseudo-pregnant mother, okay? And then they will give birth to multiple babies, okay? And these transgenic organisms will now be born with that particular gene of interest inside their DNA. Remember, because we injected the DNA into the embryo at its very earliest stage possible, every single one of those cells within these transgenic mice should have the same inserted DNA sequence because they came from that one embryo cell that had the DNA of interest, okay? So usually 1 out of several hundred embryos actually becomes a transgenic organism, and the integrated DNA, like I said, will be in every single cell of that transgenic organism. And also when that transgenic organism is to mate with another mouse, for example, they will pass that gene to the next generation because its gametes also has that particular gene. Whether it's sperm or egg, it will still contain that same gene that you have inserted. Okay, so even though it takes a long time to develop, once you have one, you're bingo. Okay, ta-da, you're done. Okay, then you're gonna just keep mating that same transgenic animal to keep having that transgenic animal there. Okay, all right. So this is my experience from my own undergraduate research. I was lucky enough to be able to work at a diabetes research institute for 2 years, and they were developing a type 2 diabetes model mice. These were called DBDB mice, where a gene has been knocked out of them, where their leptin receptor has become non-responsive. Okay, if you remember from anatomy and physiology that the leptin receptor is critical for your sensation of being full, meaning after you eat, you feel full because you have leptin as your hormone and you have leptin receptor in your brain that tells you, hey, you better stop eating because you have food in your stomach and you're full. Well, to mimic type 2 diabetes, which is a case where usually it's obese individuals where they have a problem with eating and accumulating a lot of fat in their body, that's what they're doing to these mice, is knock out that leptin receptor so that these mice actually ends up eating and eating and eating in their cage. They don't stop eating because they don't know when they're full, and hence that's why you see me here holding a fat little mouse in my hand, because technically speaking, normal little mouse will pounce around and dance around and jump around the moment you even touch it. It won't sit in your hand like that. But because of how big these mice are, they are kind of, stable and calm. Okay, so this is how big the type 2 diabetes mouse model would look like versus a normal adult mouse. Okay, usually they're almost 3 or 4 times the weight. Usually a normal mouse weighs around 15 to 20 milligrams. These type 2 diabetic mouse actually weighs about 50 milligrams when they become an adult. Okay. So, and it did took them 1 to 2 years to develop these diabetic mouse. But what we were studying there then is not just for the fun of creating these transgenic mouse. We were trying to see whether knocking in an extra gene would actually rescue these type 2 diabetic mouse from becoming diabetic. Okay, we would actually knock in 2 genes, the diabetic gene and a rescue gene that technically should reverse the effects of diabetes, and hopefully the mouse would not develop diabetes and their blood glucose would be normal. So that was the goal of that research. Okay, so a lot of these research with transgenic animals is hopefully we can test something, that a concept that we think should hold true in humans, and we can test it in a mouse before doing any clinical trials on human beings. All right, so again, this diagram here shows why mouse is used as a model organism in genetics. Okay, note that there is 99% sharing of genes between mouse and humans, and that's why we want to use them as our model organisms. They have rapid reproduction and they are small size, so we can easily keep them, and it actually tolerates inbreeding. So we can actually mate siblings together, and we can mate transgenic animals with transgenic animals, etc., to keep the transgenic line going. Okay, and part of my job in that research project was to keep the transgenic line going by keep breeding in between transgenic animals that were siblings from that one pair of mouse. Okay, so the last topic the chapter ends with is called gene therapy. So gene therapy, which is usually the ultimate goal and application of recombinant DNA technology we all hope to achieve for all genetic related diseases one day. To current day, the vehicle they're trying to use for transferring a gene into humans are viruses, because viruses are small and some of them have the capability of integrating their DNA into our genome. If we can make sure that the viruses being used as gene carriers won't cause a disease, then this would be an efficient route for delivering a gene. Note that this is different from taking medication from over the counter. We're not trying to deliver a small, tiny little chemical into the circulatory system and be absorbed by a specific organ. It is much more complicated than that. We are trying to deliver an actual gene into a cell's nucleus and hopefully swap out a mutated gene with a normal gene. So this is kind of like putting it into a nutshell right here in this picture. We have a healthy gene and we're putting into a virus vector. And this virus vector with the healthy gene hopefully can infect the person's cells. And that virus hopefully can integrate that healthy gene back into the genome of this person. And that person will be cured of the disease. Okay? Again, easier said than done. It's a very, because there's actually lots of secondary symptoms and also side effects that we don't expect to happen, but it sometimes happens. The two viral vectors they are using as gene carriers are called adenovirus herpes simplex virus or retroviruses. For adenoviruses or herpes simplex virus, these will remain as an episome, meaning that they will not integrate the specific healthy gene into the chromosome or genome of the individual. So the gene expression is actually transient. It's a temporary effect, okay? But maybe if that's what we can do, at least we can do that. But, a better route is use retroviruses, which integrates a perfect your normal version of the gene into the chromosome or the genome. This offers more potential for long-term expression and can only integrate, unfortunately, in dividing cells because this particular retrovirus cannot enter the nuclear membrane. Okay, remember where our genome resides in our nucleus of the cell. In order to fix our mutations within our genome, that particular viral DNA gotta enter into our nucleus. And hence, these retroviruses cannot enter the nucleus. So unless the cell is dividing where the nuclear envelope actually breaks apart during mitosis, then you can never have the DNA going into your genome. Okay, so but in dividing cell, it has a chance because that nuclear envelope falls apart. And currently, the gene therapy has been attempted to treat somatic cells only, meaning that cells that has nothing to do with your germline, okay? Not your sperm cells or your egg cells, but your organ cells, your skin cells, your hair cells, whatever it is, okay? We call that somatic gene therapy. Now, technically speaking, the same thing can be done with germ cells, but there can be some ethical concerns if we're talking about germline gene therapies because then we're technically manipulating the gene pool of future generations. What if the gene therapy didn't do what it's supposed to have done and ends up introducing cancer to all the germ cells? That's intense. Very bad, effect for everything or all the children. Or more extreme cases of foreseeing what can be done if germline gene therapy ever becomes successful is would people try to use the same method to enhance certain human traits, such as good looks, height, advantages, intelligence or athletic ability, have bigger muscles or better running technique, abilities to win Olympics and marathons, This was the whole idea of eugenics, and hence that is why germline gene therapy is still not, sort of common because it just kind of goes into the gray area in terms of human ethics. Now, there's not much of a history of gene therapy because of its potential danger and side effects that we don't know if it will occur in each individual. And there have been cases in history where 1 or 2 out of a handful of clinical trials of gene therapy have gone haywire, and either they resulted in death of the individual, or led to cancer. Here are a few cases of gene therapy in the past. In 1990 was the first ever case of SCID. Remember, SCID is a severe combined immunodeficiency in a child. Her name is Ashanti De Silva. I think she was 4 years old when she was cured in 1990 of her SCID disease due to gene therapy. And this was her going through gene therapy once, and supposedly she is still alive. And it has been 20 years since her at least the article that I was reading, it has been 20 years since her gene therapy, and they still found that 25% of her T cells are still active in her immune system after that long. So that one therapy did its job. In 1999, however, they were trying to use similar gene therapy techniques to cure another disease called ornithine transcarbamylase OTC deficiency, known as OTC deficiency. It's a type of metabolic disease that's linked to your X chromosome, and it is specifically a disease of the liver. So this person named Jesse Gelsinger actually was one of the many patients that have undergone this clinical trial for gene therapy for this disease. Just 4 days after the gene therapy, he unfortunately died of multiple organ failure due to a massive immune reaction following the injection of the gene therapy virus. And this particular clinical trial dosage of engineered adenovirus to treat this disease. Other patients were just fine. There were other participants in the clinical trial. They had no problems, but he was the one that had a really bad reaction and ended up dying pretty soon. So that's why we say gene therapy, we never know what happens in different individuals. That's why it's still not a widely used way to cure genetic diseases, because we don't know everything yet. And in 2000 and 2001, 10 out of 12 patients were cured of SCID. This was again somewhere in Europe. They were very happy. Everyone was very excited about this, including the case in 1990. But in 2002, 2 of those 10 cured SCID patients had ended up developing leukemia as a result of gene therapy. A possible cause that have came up with is that maybe the virus also integrated next to an oncogene. Remember, oncogene means a gene that's likely to cause cancer in some cells and ends up being cancerous in a few patients. And because we're injecting or infecting specifically blood cells in these SCID patients, they end up with blood cancer, which is leukemia. Currently, the United States has not approved many of these gene therapies. There's some, but just not a whole bunch of them, like normal clinical trials. But many are in FDA trials. You can go ahead and look at some of these in clinicaltrials.gov for gene therapies that are currently under clinical trials. This last slide here goes over how gene therapy is carried out for SCID patients. Is actually quite ingenious as well. You have your genetically disabled retrovirus, which it does not contain the viral genome, and you would actually insert the particular plasmid with the clone normal version of what's called the ADA gene, because in some SCID patient, they actually have a mutation within the ADA gene. So they put in this correct ADA gene, the wild type version, into the virus. So this virus is just an empty pocket with that particular wild type version of the gene we want to put into these patients. And T cells were harvested from SCID patients. The first case, they harvested T cell, and later on they moved on to harvesting hematopoietic stem cells. Remember from anatomy that HSC cells are those stem cells that reside in your bone marrow for construction of any red blood cell and white blood cell that's floating around your body. So if it's your blood stem cells that they're correcting, then they can correct you for life. Okay. So other than T cells, HSC cells are also the other type of cells that's being infected with this particular retrovirus method. And they would put those white blood cells with either HSC or T cells back into the baby with that corrected gene, and hopefully these cells would be enough to cultivate and start to have a colony of cells with the correct version of the gene and rebuild their immune system from the beginning, from scratch. Okay. All right, so this is a very good technique because technically you won't have a rejection problem. These are your own cells that's harvested from the body. We just put a gene in there that corrects your mutated gene, and then we put those corrected version of the cells back into the baby itself. Okay, so it's, very straightforward and safe technique, technically speaking, but you never know what this virus do inside different people's bodies. Sometimes it can lead to immune effects, or there can be wrong integration of the gene, and it can activate an oncogene in those leukemia patients. Okay. All right, so this concludes chapter 14, again, which is a very informative, heavy chapter, a long chapter, which is why we're not going to do chapter 15. I will explain that in my email. We will wrap up the semester next week with chapter 16 on cancer genetics, which is also another super interesting topic. Okay, and we'll talk about that next week. Now, let's go over some of those practice questions for this chapter, and we'll be done. Which of the following is not a required step in genetically engineering bacteria that can produce a human protein? Insert human protein in the bacteria, because that's what we're relying on the bacteria to do for us, is to make the protein, not us putting the protein into the bacteria itself. So all of these are true other than this one. Restriction enzyme A has recognition site this, okay, which is made up of 6 nucleotides. Restriction enzyme B has recognition site that's this, which is just 4 nucleotides. Based on this information, you can infer that. Remember what I said about length of recognition sites, okay? The longer recognition site, the less DNA fragments you have. So hence, enzyme A will cut the genome into fewer pieces than enzyme B, because enzyme A has a recognition sequence that's longer than enzyme B. Which of the following is not true regarding DNA gel electrophoresis? Larger DNA fragments will not migrate farther. It is the smaller DNA fragments that will migrate further. All of these other ones are true for gel electrophoresis. Which of the following is not required for PCR? DNA ligase. Okay, DNA ligase was important for gene cloning. Okay, for putting our insert of foreign DNA into our cloning vector, but not important for PCR. PCR, we're just amplifying our target sequence. The discovery of PCR has revolutionized molecular biology studies. However, there are limitations to PCR. Which of the following is a limitation? Part of the target DNA sequence needs to be known, because what do we need to design in PCR? Primers, okay? We need to design those forward primers and reverse primers, and hence we have to know partially the sequence of our gene of interest to be able to design those primers and buy those as little oligonucleotides from companies, so we can have those primers to start the amplification process for us. Gene cloning requires placing the gene of interest into a cloning vector. A good cloning vector must have which of the following? All of these 3 things, okay? You are studying a human gene code encoding a protein that requires post-translational modification. What feature is or are important in picking your vector? Both A and B. It needs to be an expression vector, because this is talking about post-translational thing, and it needs to be a vector that can replicate in eukaryotic cells, because prokaryotic cells, bacteria, cannot do post-translational modification. If you are interested in identifying genes expressed in cancer cells, why might you choose to construct and screen a cDNA library instead of a genomic DNA library? A cDNA library is enriched for genes that are actively transcribed in your cell of interest. Remember, I said cDNA library is a more direct way of looking for gene that actually makes products, okay, protein products. So that's why cDNA library can be more efficient in that sense. When present in small amounts in sequencing reaction, ddNTPs terminate sequencing reaction in different positions in the growing DNA. ddNTP stops the sequencing reaction because they lack a hydroxyl group at their 3' end, so DNA polymerase doesn't go on. It requires a free 3' hydroxyl group. Compared to the original Sanger method of DNA sequencing, next generation sequencing methodologies, both A and B is true. Sequencing is faster and allows hundreds of thousands of millions of DNA fragments to be simultaneously free sequenced compared to the original first generation, the Sanger method. Gene therapy involves transferring desired genes into human cells to treat disease. Which of the following is not true about this technology? It involves transfer of genes into germline cells. No. Remember, we talked about how germline cell gene therapy is something we don't want to touch yet due to the gray area of ethical concerns. Okay, these other things are true about gene therapy. And that's all we have. All right, see you